Human DNA unzips at 175 degrees Fahrenheit. That is cooler than boiling water. The molecule that contains every instruction for building your entire body, your height, the color of your eyes, the shape of your hands, comes apart at the temperature of a hot cup of coffee. The double helix separates. The genetic code becomes unreadable. And that is just the beginning of the destruction. Modern cremation chambers reach 1800 degrees Fahrenheit. At that temperature, steel glows red. Organic molecules vaporize. Bone becomes powder. Everything that made you physically you is supposed to be gone. And yet, when forensic scientists examined cremated remains, they found fragments of DNA still intact. Not enough to say who you were, but enough to prove you were once alive, enough to prove you were human. The most fragile molecule in your body outlasted a fire that turned your skeleton to dust. The question is how? How does a molecule held together by the weakest bonds in chemistry survive conditions that destroy almost everything else? And the answer involves a shield that is 300 million years old. You see, to understand how anything survives cremation, you first have to understand how easily DNA falls apart. And it falls apart very easily. In 1953, two scientists at Cambridge, James Watson and Francis Crick, figured out the structure of this molecule. They built a physical model out of metal and wire, and what they found was beautiful. Two long strands twisted around each other in a spiral, a double helix. Each strand is a chain of smaller molecules, nucleotides, and the two strands are held together by bonds between those nucleotides, the famous base pairs. Adenine pairs with thymine, guanine pairs with cytosine, and the bonds holding those pairs together are hydrogen bonds. Now, here is the thing about hydrogen bonds. They are among the weakest bonds in all of chemistry. A single hydrogen bond has roughly 1/20th the strength of the covalent bonds that hold together the backbone of each strand. 1/20th. This is the bond that holds your genetic code in its readable, double-stranded form. And it is so weak that warm water can break it. Think about that. The information that specifies everything about your biology, how to build your brain, how to grow your bones, how to repair a cut on your finger, all of that information is held in place by bonds so delicate that a fever could start to disrupt them. When your body temperature rises even a few degrees above normal, some of your DNA begins to denature. The strands start to separate. Your cells have repair machinery that fixes this constantly, every minute of every day. But the molecule itself is startlingly fragile. At around 200 degrees Fahrenheit, DNA denatures completely. The double helix unzips into two single strands. The hydrogen bonds are gone. The elegant spiral Watson and Crick built out of metal and wire, that structure ceases to exist. And at this point, you have not even reached the boiling point of water. You have not come anywhere close to a cremation furnace. So what happens as the temperature keeps climbing? Between 300 and 400 degrees Fahrenheit, the backbone itself begins to break. Each strand of DNA has a spine made of alternating sugar and phosphate groups. Deoxyribose sugar, phosphate, deoxyribose sugar, phosphate, linked by covalent bonds that are much stronger than those hydrogen bonds between the base pairs. But even covalent bonds have limits. At 300 to 400 degrees, those bonds start to crack. The long strand fragments into shorter pieces, then shorter still. The molecule that, in its intact form, stretches to about 6 feet of total length inside every one of your cells. 3.2 billion base pairs, shatters into debris. By 500 degrees, most of the organic components of DNA are undergoing pyrolysis. The carbon-based structures are breaking apart. The nitrogen in the bases escapes as gas. The phosphorus oxidizes. What was once a precisely ordered sequence of genetic information becomes a disordered mess of molecular fragments, most of them no longer recognizable as having once been DNA at all. And a cremation furnace operates between 1400 and 1800 degrees Fahrenheit for 2 to 3 hours. So on paper, the situation is hopeless. The molecule is gone, not damaged, not degraded, gone, vaporized, returned to its constituent atoms, Carbon dioxide, water vapor, nitrogen gas, phosphorus oxides scattered into the exhaust system of the crematorium and out into the atmosphere. There should be nothing left to find, nothing left to read. And for most of the DNA in your body, that is exactly what happens. The DNA in your skin, your muscles, your organs, your blood, it is destroyed quickly and completely. Those tissues are soft, they are full of water, they burn. The DNA inside them has no protection whatsoever, and it is gone within the first minutes of cremation. But your body is not uniform. Different tissues protect their DNA differently. And this is where the story takes its turn. You see, not all of your DNA sits naked inside soft cells. Some of it is locked away inside the hardest structures in your body, your teeth, your bones, and the reason those structures are hard, the very thing that gives them their rigidity, turns out to be the same thing that protects the DNA inside them. The mineral is called hydroxyapatite. It is a crystalline form of calcium phosphate, and it is the primary mineral component of bone and tooth enamel. Your skeleton is not solid calcium. It is a composite material, a matrix of collagen fibers and hydroxyapatite crystals woven together. And your teeth, particularly the enamel, are even more heavily mineralized. Tooth enamel is about 96% hydroxyapatite by weight. It is the hardest substance your body produces. Now here is what makes this relevant. Hydroxyapatite does not just give your bones and teeth their structure, it forms a crystalline cage, a mineral lattice that physically surrounds and encases the DNA molecules embedded within it. The DNA in your bone cells and tooth pulp is not floating freely in liquid cytoplasm like the DNA in your liver or your lungs. It is trapped inside a rigid mineral framework, locked in crystal. And this was the shield I mentioned, 300 million years old. Because hydroxyapatite is not a recent invention. Vertebrates have been building their skeletons out of this mineral since before the first animals walked on land. It evolved to give fish their bones, it evolved to let amphibians support their weight out of water, it evolved to let you stand upright. And entirely by accident, as a pure side effect of structural engineering, it created a microenvironment that partially shields DNA from destruction. The mineral matrix does two things. First, it physically blocks oxygen from reaching the DNA. Combustion requires oxygen. When organic molecules are exposed to high heat in the presence of oxygen, they combust, they burn, they oxidize, they are converted to gases and ash. But inside the mineral matrix of bone, the oxygen supply is restricted. The DNA does not combust. Instead, it chars. And that distinction matters enormously. Charring is what happens when organic material is heated in a low oxygen environment. The molecules do not burn completely to carbon dioxide and water. Instead, they partially decompose, leaving behind a carbon-rich residue. Think of it like the difference between a log burning to white ash in an open fire versus a log smoldering into charcoal in a sealed kiln. The charcoal still contains carbon structures. It still has molecular information, however degraded. Complete combustion leaves nothing. Charring leaves something. The second thing the mineral matrix does is slow the transfer of heat. Hydroxyapatite is a poor thermal conductor. It acts as insulation. The interior of a dense bone or a tooth does not reach the same temperature as the surface of that bone or tooth, at least not immediately. During cremation, the outermost layers of bone are exposed to the full 1400 to 1800 degrees, but the deepest interior regions, particularly in the densest bones, the femur, the petrous bone of the skull, the roots of the teeth, experience lower peak temperatures for at least part of the process. The thermal gradient buys time, and time, at these temperatures, is the difference between total destruction and partial survival. This is how DNA fragments persist in cremated remains. Not intact DNA, not readable genomes. Fragments. 50 to 100 base pairs long, compared to the 3.2 billion base pairs of a complete human genome. Tiny shards of what was once the longest molecule in your body, but fragments that are unmistakably DNA. Fragments that carry enough sequence information to confirm that they came from a human being. Think about that for a moment. The molecule unzips in warm water. It fragments by 400 degrees. And yet pieces of it survive 2 to 3 hours at 1400 degrees, locked inside the mineral skeleton that evolution built for an entirely different purpose. Now, everything I have just told you about bone and mineral shields and charring, that is the chemistry, and it is beautiful chemistry. But there was a group of scientists who needed this chemistry to do something very specific. They needed it to identify the dead. In 2001, a researcher named Michaela Harbeck and her colleagues published a study in the International Journal of Legal Medicine. What they attempted was something many people in the field considered a waste of time. They took cremated remains, real cremated remains from real cremation facilities, and they tried to extract DNA from them. Now, you have to understand what the scientific consensus was at the time. Cremation was considered the end of the line. If a body was cremated, the DNA was gone, period. Forensic scientists had well-established protocols for extracting DNA from decomposed bodies, from skeletal remains, from bodies recovered from water, from fire-damaged remains in house fires and airplane crashes. But cremation, deliberate, prolonged, industrial temperature cremation, was assumed to be beyond recovery. The temperatures were too high. The duration was too long. There was nothing left to work with. And this was not just an academic question. It was a forensic problem with real consequences. Because some people cremate bodies precisely to destroy evidence. If a person disappears and no body is found, there is no case. If remains are found but cannot be identified, the case goes cold. Cremation was understood in forensic circles as the most effective way to erase biological identity. The fire does what time and decay would take centuries to accomplish. It does it in an afternoon. So Harbeck and her team took their samples and they attempted extraction. And they found DNA. Damaged, degraded, fragmented, but present. Short sequences, 50 to 100 base pairs, recovered from the densest bone fragments that survived the cremation process. Fragments from the interior of bones where the mineral matrix had done its work, where charring had occurred instead of complete combustion, where the thermal gradient had kept temperatures just low enough for just long enough. But here is where the story gets complicated. Because finding DNA and identifying a person from that DNA are two very different things. The standard method for forensic identification is called STR analysis, short tandem repeat analysis. This technique looks at specific locations in the genome where short sequences of DNA repeat over and over. The number of repeats varies from person to person, and by examining enough of these locations, you can build a genetic profile that is unique to an individual. It is the basis of every forensic DNA database in the world. It is what links a suspect to a crime scene. It is what identifies remains after disasters. But STR analysis needs DNA fragments that are at least 200 base pairs long. 200. And cremation leaves fragments of 50 to 100. The pieces are too short. The genetic signature is too shattered. You cannot run a standard forensic identification on cremated remains because the fragments that survive are smaller than the minimum length the technology requires. So the answer to the forensic question, can you identify a murder victim from cremated remains, is, in most cases, no. The DNA is there. You can confirm it is human. You can sometimes determine biological sex. But you cannot generate an individual profile. You cannot match it to a missing person. You cannot put a name to the ash. And this is where most people think the story ends. Cremation wins. The fire erased the identity. Case closed. But there was one type of DNA they had not tried yet. A different molecule, carried differently, inherited differently. And it changed the answer. You see, when we talk about DNA, most people picture the nuclear DNA, the double helix in the nucleus of the cell, the 3.2 billion base pairs, the 23 pairs of chromosomes. That is the DNA that STR analysis targets. That is the DNA that is unique to you as an individual. And that is the DNA that cremation destroys beyond forensic usefulness. But there is another kind of DNA in your cells, and it lives somewhere else entirely, inside your mitochondria. the tiny structures that generate energy for your cells, there is a separate, much smaller genome, mitochondrial DNA, and it is different from nuclear DNA in almost every way that matters for this story. First, it is small. The human mitochondrial genome is only about 16,500 base pairs long compared to 3.2 billion for the nuclear genome. It is a tiny circle of DNA, compact and dense with information. Second, and this is the critical part, there are not just two copies of it per cell, there are hundreds, sometimes thousands. Every mitochondrion in every cell contains multiple copies of the mitochondrial genome, and every cell contains hundreds or thousands of mitochondria. Your muscle cells might contain 2,000 mitochondria each, your heart cells even more. This means that for every two copies of nuclear DNA in a cell, there might be 1,000 or 5,000 copies of mitochondrial DNA. Think about what that means for survival. If cremation destroys 99.9% of the DNA in a bone cell, and there were only two copies of nuclear DNA to begin with, you are left with nothing. But if there were 5,000 copies of mitochondrial DNA and 99.9% are destroyed, you still have 5 copies left. The sheer redundancy, the massive copy number, gives mitochondrial DNA a survival advantage that nuclear DNA simply does not have. And the third thing, mitochondrial DNA is inherited only from your mother. Every copy in your body is identical to every copy in your mother's body, and her mother's body, and her mother's body before that. It does not recombine. It does not shuffle. It is a direct maternal line stretching back through generations. So when forensic scientists extracted mitochondrial DNA from cremated remains, they found something they could work with. The fragments were still short. The damage was still severe. But because the starting copy number was so much higher, there was more material to recover. And because the sequences needed for maternal lineage identification are shorter than those needed for individual STR profiling, the fragments that survive were sometimes long enough to be useful, not always, not reliably, not enough to build a legal case in most jurisdictions, but sometimes, in some cases from some remains, when the densest bones provided the best shielded fragments, forensic scientists could extract enough mitochondrial DNA to determine a maternal lineage, not a name, not a face, not a unique identity, but a family line, a connection, a thread leading back to a mother, a grandmother, a great-grandmother. You cannot say this person was John, but you can sometimes say this person belonged to the same maternal line as this family. And in a missing persons case, that can be enough to change everything. Now, I want you to hold that in your mind. This image of forensic scientists extracting fragile molecular whispers from the inside of cremated bone, because there is a contrast I need to show you. And it is the contrast that makes this entire story land differently. There was a scientist named Svante Pääbo who spent decades working on a seemingly unrelated problem. Starting in the 1980s, working first in Uppsala, Sweden, and later in Munich and Leipzig, Pääbo was trying to extract DNA from ancient remains. Not cremated remains, buried remains, buried remains. Bones and teeth recovered from archaeological sites thousands of years old. And what he found was astonishing. DNA, readable, sequenceable DNA, surviving inside the teeth and bones of humans who had been dead for 10,000, 20,000, 40,000 years. The petrous bone of the skull, one of the densest bones in the human body, turned out to be a remarkable natural vault for genetic material. Teeth, sealed by their enamel, were another. In the cold, dry, oxygen-poor environments of certain burial sites, DNA persisted not for decades or centuries, but for millennia. Pääbo's work eventually led to the sequencing of the entire Neanderthal genome, a species that has been extinct for roughly 40,000 years. Think about that. 40,000 years of burial and enough DNA survived in bone to reconstruct a complete genetic blueprint of a vanished human species. His work launched the entire field of ancient DNA research and earned him the Nobel Prize in Physiology or Medicine in 2022. And here is the contrast that should stop you cold. Burial preserves DNA for 40,000 years. Cremation destroys almost all of it in 2 hours. The same mineral shield, hydroxyapatite, protects DNA in both cases. The same bones, the same teeth, the same crystalline cage of calcium phosphate. But in burial, the shield holds. In cold ground, in dry caves, in permafrost, the mineral matrix keeps oxygen out, keeps enzymes away, keeps the slow chemical degradation of DNA to a pace so gradual that readable fragments persist across geological timescales. In cremation, the shield slows the destruction. It buys minutes, maybe an hour, in a furnace that runs for 2 to 3 hours. It converts combustion to charring in the deepest bone. It preserves fragments, tiny, degraded, barely readable fragments that would not exist at all without it. But it cannot hold against 1800 degrees for 3 hours the way it holds against 50 degrees for 50,000 years. Fire does in an afternoon what time takes millennia to accomplish, and it does not do it completely. That is the strange part. You would expect one or the other, total destruction or significant survival. Instead, you get this, almost nothing, but not nothing. Fragments too small to identify an individual but large enough to confirm a species. Traces too degraded for a courtroom, but sufficient for a family line. The code is shattered, but the alphabet is still there. You know, there is something I find deeply interesting about this. We think of cremation as erasure, as the most thorough destruction of a body that ordinary technology allows. And in almost every practical sense, it is. The water is gone. The carbon is gone. The carbon is gone. The proteins, the fats, the carbohydrates, all gone. What remains is calcium phosphate powder and fragments of mineral bone. The physical person is, by any meaningful measure, absent. But the molecule that defined that person, the one that specified every protein their cells ever made, the one that carried the instructions their mother's egg and their father's sperm combined to create. That molecule does not disappear entirely. Pieces of it, locked inside the mineral architecture of bone, persist. Charred remnants, protected from the oxygen that would have finished the job, endure. They are unreadable as an identity. They are insufficient as evidence. But they are not nothing, and I think there is a profound asymmetry here that deserves attention. If you bury a body, the DNA inside the teeth and bones of that person can survive for 10,000, 20,000, 40,000 years, long enough for an entirely different civilization to dig it up and read it. Long enough for scientists to determine not just that this person was human, but who their relatives were, what diseases they carried, what color their hair was, where their ancestors migrated from. Burial preserves genetic identity across timescales that dwarf a human lifetime. If you cremate a body, the DNA is reduced to unidentifiable fragments in a single afternoon. The mineral shield that held for millennia in the ground holds for minutes in a furnace. The information is not preserved. The identity is not recoverable. The fire compresses into hours a degradation that nature, left to its own patient processes, would spread across geological time. And yet, even after that fire, something remains. Not you, not your identity, not anything a court or a database or a grieving family could use to say, yes, that was them, but a molecular trace, a chemical signature, a fragment of the code that once specified a living, breathing human being. 50 base pairs out of 3.2 billion. A whisper where there was once a symphony. Nature uses only the longest threads to weave her patterns, so each small piece of fabric reveals the organization of the entire tapestry, and even the smallest fragment of DNA. 50 base pairs recovered from the charred interior of a cremated femur still carries the pattern, still uses the same four-letter alphabet, still pairs adenine with thymine, guanine with cytosine, still obeys the rules that Watson and Crick discovered in 1953 when they built their model out of metal and wire and called it beautiful. The structure is gone, the helix is unwound, the information that made you specifically you is scattered beyond recovery, but the language itself persists. The molecular grammar that life has used for billions of years to encode every organism on this planet, from bacteria to blue whales to the particular human being whose remains now sit in an urn on someone's mantelpiece. That grammar is still legible in the fragments. You try to erase yourself with fire, and the code that built you leaves a trace, not enough to reconstruct you, not enough to name you, but enough to say unmistakably that something was here, that something was alive, that something was human. Remember the question from the beginning. How does the most fragile molecule in your body outlast the fire that turns your bones to powder? The answer is that it does not, really. Not in any way that preserves who you were. The fire wins. The identity is gone. But the molecule, the thread of nucleic acid locked inside a mineral cage that evolution built for an entirely different purpose 300 million years ago, does not surrender completely. It chars instead of burning. It fragments instead of vaporizing. It leaves behind 50 base pairs in the deepest bone, a residue so faint that only the most sensitive instruments in a forensic laboratory can detect it. And those 50 base pairs, degraded and anonymous as they are, carry something remarkable. They carry proof. Not proof of identity. Proof of existence. Proof that the four-letter code of life was once running in this body, in these bones, in this person. The fire took the message, but it could not take the language. And that, I think, is as close to immortality as a molecule gets.